Application of immunological principles, vaccine diagnostics. A vaccine is prepared of microorganisms or some part of product of them that will induce an immune response when injected into a host. The practice of vaccination, like many medical practices, predates our understanding of the process. Many culture affiliate with the smallpox adopted the practice of intentionally expose themselves to material from smallpox lesions. If successful, this leads to a mild infection followed by immunity, and if unsuccessful, it could be led to severe infection and even death. In the 18th century, Edward Jenner established a safer method for vaccination against smallpox. Jenner observed that persons that had been exposed to cowpox, which causes a mild infection in humans, exhibited immunity to smallpox. The Jenner began deliberately exposing patients to materials from what was thought to be cowpox lesion in the first controlled vaccination trial. Eventually, these efforts led to one of the greatest public health victories in history, as there are complete elimination of smallpox from human populations. The vaccines are critically important for control of infectious disease, for which other preventive efforts and or antimicrobial drugs are inadequate. This is especially the case for infectious disease caused by viruses. The goal of vaccination efforts is to provide herd immunity to a population so that if an outbreak should occur, an epidemic cannot develop because there are not enough susceptible hosts. The vaccine may be of several types depending on the target pathogens. The inactivated vaccines are preparations of killed microorganisms. The original Salk polio vaccines was on inactivated vaccines. The attenuated vaccine, of which Jenner vaccine was an example, or viable but so weakened as to be non-pathogenic. An advantages of attenuated vaccine is that they may cause a limited infection that invokes a stronger immune response than an inactivated vaccine preparation. The attenuated vaccines are currently used for polio, measles, rubella, and mumps. The subunit components vaccines employed part of the viruses or bacterium. The highly recommended vaccines for hepatitis B virus consist of one of the capsid protein of the viruses. The capsule or polysaccharide are used as a vaccine against infection with the Streptococcus pneumonia, agent of bacterial pneumonia, and Haemophilus influenza, the agent of bacterial meningitis. The capsule or polysaccharide are used as a vaccine against infection with the Streptococcus pneumonia, the agent of bacterial pneumonia, and Haemophilus influenza, agent of bacterial meningitis. The the toxoids are denatured bacterial toxins. The toxoids are used in vaccination against tetanus and diphtheria. The vaccine administration does not carry some degree of risk. In very rare cases, an attenuated vaccines may mutate to a pathogenic form or cause a pathogenic infection in a severely immune compromised host. This has been recorded for the Sabin polio vaccine. There may be a negative reaction in some individuals. This is at the root of concern about the liability for pertussis vaccine. The advances in molecular biology are presently considering to vaccine development. The attenuated vaccine can be analyzed and manipulated in effort to avoid the possibility of reversion. The subunit vaccine can be prepared by genetic engineering. This is already the case for the hepatitis B vaccine, which is synthesized by yeast cell DNA. Vaccines are currently received a lot of attention. The idea here is that the gene will be expressed in cell and invoke a cell-mediated immune response. Compared with the whole and protein vaccines, the DNA offers tremendous advantages in cost and stability. The diagnostic immunology employs the antigen-antibody interaction on to identify the presence of antigen or antibody in test material. The serology detection and measurement of specific antibody in the patient's serum is often important in identifying infectious agent, particularly as in most viral infection when isolation of agent is difficult. The precipitation reaction involves formation of large aggregate of antigen and antibody. Such aggregate forms only when there is an optimal ratio of antigen antibody. A variety of technique including diffusion of antigen and antibody through agar gels can be used to visualize the characteristics precipitating, whereas precipitation reaction involves soluble antigen agglutination reaction involving antigen bound to particles. Application of immunological principles. One precipitation reaction, the antibody and a soluble antigen interaction is aqueous in aqueous solution form a lattice that eventually developed into a visible particles precipitate. Antibody that aggregate soluble antigen are called precipitin. Although the formation of a soluble antigen antibody complex occurs within minutes, the formation of visible precipitate occurs more slowly and often takes a day or two to reach completion. To agglutination reactions, the interaction between the antibody and a particular antigen results in visible clumping called agglutination. The antibody that produces such a reaction are called agglutination. The agglutination reaction are called agglutinins. The agglutination reaction are similar in principle to precipitation reactions. They depend on the cross-linking of polyvalent antigen. Just as an excess of antibody inhibits precipitation reactions, such excesses can also inhibit agglutination reaction. This inhibition is called the zone effect. 3. Radio immunoassay, one of the most sensitive technique for detecting antigen or antibody in radio immunoassay RIA. This technique was first developed in 1960 by two endocrinologists S.A. Berson and Rosalind Yalow to determine level of anti-insulin complexes in diabetes. Although their techniques encountered some skepticism, it soon provided its value for measuring hormones, serum proteins, drugs, and vitamins at a concentration of 0.001 microgram per milliliter or less. 4. Enzyme-linked immunosorbent assay ELISA. The ELISA, enzyme-linked immunosorbent assay, commonly known as ELISA or EIA, is similar in principle to RIA but depends on an enzyme rather than a radioactive label. An enzyme conjugated with an antibody reacts with a colorless substrate to generate a colored reaction product. Such a substrate is called chromogenic substrate. A few examples have been employed for ELISA, including alkaline phosphatase, horseradish peroxidase, and beta-galactosidase. These assays approaches the sensitivity of radioimmunoassay and have the advantages of being safe and less costly. 5. Western blotting. The identification of specific protein in a complex mixture of protein can be accomplished by a technique known as Western blotting, named for its similarity to Southern blotting, which denotes DNA fragments, and Northern blotting, which detects the mRNA. In Western blotting, a protein mixture is electrophoretically separated on an SDS polyacrylamide gel, SDS phase, a slab gel infused with a sodium dodecyl sulfate, a dissociating agent. 6. Immunoprecipitation. The immunoprecipitation technique has the advantages of allowing the isolation of a antigen of interest for further analysis. It also provides a sensitive assay for the presence of antigen in each cell or tissue type. An extract produced by disruption of cell or tissue is mixed with an antibody against the antigen of interest in order to form an antigen antibody complex that will precipitate. However, if the antigen concentration is low, often the case in cell and tissue extract, the assembly of antigen antibody complexes into precipitate can take hours, even days, and it is difficult to isolate the same amount immunoprecipitation that forms. 7. Immunofluorescence. In 1944, Albert Kunz showed that antibody could be labeled with molecules that have the property of fluorescence. The fluorescent molecule absorbs light of one wavelength excitation and emits light of another wavelength emission. If antibody molecules are tagged with the fluorescent dye or fluorochrome, the immunocomplexes containing these fluorescently labeled antibody FA can be detected by colored light emission when excited by light of the appropriate wavelength. 8. Flow cytometer and fluorescence FACS. The fluorescent antibody technique described are extremely valuable qualitative tools, but they do not give quantitative data. This shortcoming was remedied remedied by development of flow cytometer, which was designed to automate the analysis and separation of cells stained with fluorescent antibody. The flow cytometer used a laser beam and light detector to count single intact cell in suspension. 9. Electron microscopy. The fine specificity of antibody has made them powerful tool for visualizing specific intracellular tissue component by immunoelectron microscope. In this technique, an electron dense label is either conjugated to the Fc portion of a specific antibody for direct staining, or conjugated to an anti-immunoglobulin reagent for indirect staining.